Chapter 11, Cell Communication. The first step in signal transduction pathways is reception. Ligands are signals that cannot pass through the cell membrane. So, they need receptors to enter the cell. When the ligand forms the receptor, it creates a lock and key bind. In many cases, receptors are G proteins. Receptors consist of subunits that are attached at the bottom of it. Adenylyl cyclase is an enzyme with regulatory functions. Step 2, transduction. When the receptor is activated, the alpha subunit attaches to the adenylyl cyclase, activating it. Step 3, messengers. After it's activated, adenylyl cyclase releases ATP, which is transformed into CAMP. The CAMP then goes to the protein kinase, which is consistent of the catalytic and regulatory system. The catalytic portion speeds up the reaction, creating the products, while the regulatory portion cuts off the catalytic portion once the activation is broken. The CAMP attaches to the regulatory portion and tears it away from the catalytic portion. The remaining ATP in the cell then is absorbed by the catalytic portion, creating an energy cascade and thus phosphorylizing. The phosphorylated catalytic kinase is then absorbed by the enzyme, activating it. Signal transduction pathways are found all throughout biology. Now let's see a real-life example.
Here we have a signal molecule trying to enter a cell through a receptor protein. But wait, it can only enter if that protein has the correct receptor to respond to the signal. There we go. Now the signal can enter, and this interaction will modify the signal molecule into the new and improved relay molecule. No! Now there's a sad inactive protein kinase that needs to be activated. In order to continue the signal pathway, the relay molecule has to bind with the inactive protein kinase. Hey, protein kinase 1 has been activated. Active PK1 transfers a phosphate from ATP to inactive PK2. Now activated PK2 does the same to inactive PK3. PK3 has been activated. Finally, active PK3 phosphorylates a protein by giving it a phosphate from ATP. Let me zoom in. Now for the main event. This active protein can now bring about the cell's response to the signal. That's it, folks. Thanks for watching.